In this next section, we're going to talk about the nature of neuronal communication. So as we talked about in under the topic of the action potential, we learned that action potentials are initiated by neuronal cells and travel down the axon and then trigger a stimulation in the next neuronal cell. And the connection between one neuronal cell and the next, the axon terminal of one neuronal cell and the dendrites of another neuronal cell, is called the synapse. And synapses tend to come in two types. We have what we call electrical synapses and chemical synapses. And by far what's more common are chemical synapses. And in a chemical synapse, we have the release of some kind of neurotransmitter by the what we call presynaptic cell, and that neurotransmitter stimulates what we call the postsynaptic cell. Okay. And neurotransmitters, these chemical messengers, can either cause stimulation of the postsynaptic cell or inhibition of the postsynaptic cell. And the release of neurotransmitter is triggered by the action potential that penetrates the axon terminal. Okay, so we'll look at that in just a moment. So the different types of neurotransmitters that are used in the chemical synapse includes things like amino acids. So many amino acids, in addition to being used as a source of energy for neuronal cells or as a in order to build proteins by that cell, some amino acids are actually used as neurotransmitters, so it's a very efficient system. We also have neurotransmitters that fall under the category of monoamines. And monoamines refers to a neurotransmitter that it is incorporated in its structure is it's derived from a single amino acid that's then been modified somehow chemically. And an example of a monoamine neurotransmitter is another category of neurotransmitters called catecholamines, and they are a group of neurotransmitters that are derived from tyrosine. Okay, and we'll see examples of that. And then finally, within the topic of chemical synapses, we also have what are called neuroactive peptides. And these are not proper neurotransmitters, so to speak, but rather they are peptides that are often co-released with neurotransmitters and modify the response in the postsynaptic cell. Okay. And so later we will talk about networks within the brain that release predominantly one neurotransmitter and supply modulatory effects on a wide variety of targets. And so some of the neurotransmitters involved in these circuits in the brain include neurotransmitters like norepinephrine, serotonin, dopamine, acetylcholine. And we will talk about that when we go through the central nervous system. Okay. And then finally, electrical synapses we will also mention. Electrical synapses are also known as gap junctions, and they actually allow for the direct spread of the action potential from the presynaptic cell to the postsynaptic cell. And we'll see what that looks like in just a moment. So the classic example of a chemical synapse is a synapse called the neuromuscular junction. And this is a synapse between the axon terminals of motor neurons. Motor neurons are the voluntary neuronal cells that stimulate skeletal muscle. So the neuromuscular junction involves the axon terminals of motor neurons and skeletal muscle. Okay, so in this picture, we have an axon terminal, a very large representation of an axon terminal of a motor neuron. You see here the myelin sheath of that motor neuron covering that axon. And then at the other end of the slide, we've got the plasma membrane of skeletal muscle. Okay? And so a few things to notice is that there is a small space that separates the presynaptic cell, which is the motor neuron, and the postsynaptic cell, which is the skeletal muscle cell. And that small space is called the synaptic cleft. Okay? And you can also tell that what's being represented here in this slide is we have those little blue circles is neurotransmitter being released by that presynaptic cell and filling that synaptic cleft and binding to receptors on the postsynaptic membrane. Okay, so let's walk through the steps of chemical synaptic transmission. So first off, you notice that what's happening up here, it looks like little explosions. We've got sodium ions entering, crossing the axonal membrane. And what that's representing is basically the action potential that has traveled down this motor neuron has eventually penetrated the axon terminal. So the action potential has traveled all the way down to the axon terminal, and this axon terminal is now firing that action potential. And what that triggers, the action potential entering the axon terminal, triggers this depolarization, right? That rapid depolarization. And that depolarization is actually going to stimulate a third voltage-gated ion channel that we haven't yet talked about, and it is the calcium-gated ion channel. So over here you see we have calcium ions that are rushing into the cell, into that axon terminal, when this terminal, as this terminal is depolarizing. Now those calcium ions, if you notice, they are entering the cell and they are stimulating the exocytosis of vesicles of neurotransmitter. Okay? And we talked about this when we talked about exocytosis. This is regulated exocytosis. That the rush of calcium into this terminal actually stimulates these vesicles of neurotransmitter to exocytose and release the neurotransmitter into the synaptic cleft. Okay? And before I complete the story, I just want to back up and show you that if you notice, these vesicles of neurotransmitter are actually they're stored within the terminal, within the axon terminal, but they are actually produced and packaged in the cell body of this neuronal cell. And they travel down the axons, right, through these microtubules within the axons, right, those superhighways. They travel down the axons and they are stored within this axon terminal. Okay, so they're packaged up in the cell body, they're shuttled down to the terminal where they sort of sit and wait to get exocytosed. So in the neuromuscular junction, this particular neurotransmitter happens to be what we call acetylcholine. So acetylcholine is being released, it's exocytosed into the synaptic cleft, and it's binding to an acetylcholine receptor on the postsynaptic membrane of the skeletal muscle. Okay? And when acetylcholine binds to its receptor on the postsynaptic cell, it actually opens another ion channel in the postsynaptic membrane. Okay? So acetylcholine binds to its receptor. It causes the opening of a cationic channel, which means an ionic channel that opens, an ion channel that allows positively charged ions to enter the cell. Okay? So positively charged ions, like sodium, now enter this muscle cell which causes a depolarization, and the depolarization, if it reaches threshold for this muscle cell, now the muscle cell is going to fire an action potential, and that's going to trigger muscle contraction. Okay? So for the neuromuscular junction, if we recap here, we have an action potential leading to the release of neurotransmitter in the presynaptic cell. The neurotransmitter binds to a receptor on the postsynaptic cell, which causes the entry of sodium, the depolarization of the muscle cell, leading to the action potential within the muscle cell and muscle contraction. Okay? So that sounds like a lot of steps, but it actually occurs very, very quickly, and it's a very common method of synaptic transmission. Okay, so that is the classic example of a chemical synapse. And so here what we have is actually a very pretty image. It's a electron micrograph image of two cells where what the arrows are pointing to are what are called active zones, and they are zones where you have the release of neurotransmitter into the synaptic cleft that sits between these cells. So you can see the synaptic cleft in this picture looks very, very, very small. And then you can see all of these little circles within the cell, which are actually vesicles of neurotransmitter. Okay? And so these darkened active zones are actually areas where those neurotransmitters are being dumped into the synaptic cleft and stimulating the neighboring cell. Okay, so different categories of neurotransmitters. We have amino acids. Now, very, very common amino acid neurotransmitters include glutamic acid, okay, which is basically glutamate. And glutamic acid is a stimulatory neurotransmitter. Okay? It is a stimulatory neurotransmitter, and it is actually the ubiquitous stimulatory neurotransmitter that we see in the central nervous system. Okay? Nearly every excitatory synapse, nearly every excitatory cell releases glutamate. Okay, so it is the general workhorse for stimulation in the brain. And then we have another amino acid neurotransmitter called aminobutyric acid, or GABA. And GABA is the generic inhibitory neurotransmitter. Okay? So whenever you have inhibition in the brain, GABA is most likely doing the job, okay? Or GABA is being released. Another inhibitory neurotransmitter that is, again, extremely common is glycine. Okay? So glycine and GABA are the two ubiquitous inhibitory neurotransmitters that we see in the central nervous system. So amino acid derived neurotransmitters include acetylcholine. We just talked about acetylcholine. Okay, serotonin derived from tryptophan, a very, very common neurotransmitter. Histamine, also very common. And then we have the catecholamines. We have these neurotransmitters, a group of neurotransmitters derived from tyrosine, and it includes, the catecholamines include dopamine, norepinephrine, and epinephrine. Okay. And then we have a category, a very, very diverse category of proteins that we call neuroactive peptides. And unlike the really large proteins that we talked about in the first lecture, neuroactive peptides are actually much shorter chains of amino acids, and they are very, very diverse. Okay, so we have a lot of familiar and not so familiar names here: somatostatin, oxytocin, endorphin. Okay. These neuroactive peptides are often released along with other neurotransmitters, especially within the brain. And what these neuroactive peptides do is they what we call modulate the response to the neurotransmitter. And they can create a variety of modulatory input to the postsynaptic cell. So, for example, a neuroactive peptide, when released with a neurotransmitter, can cause in the postsynaptic cell an increase or a decrease in the production of the receptor for the neurotransmitter. Okay. So in that case, let's say it increases the production of the receptor for the neurotransmitter. And that means that the next time that neurotransmitter is released onto that cell, that postsynaptic cell is going to create a more robust response. It's going to have a greater sensitivity for that neurotransmitter. Okay. So that enhanced sensitivity is being triggered by that neuropeptide, that neuroactive peptide. Okay. So that's what we mean by the neuroactive peptides are modulating. They're creating some sort of modulation of the response to the neurotransmitter by the postsynaptic cell. So, thank you.